Hello everyone. Today I will be talking about chromatography method. It's a common method that we have in our clinical labs. I will cover all the important points that's mentioned in TEAT's textbook. So this lecture can be a good tool, a study tool and reference. So what's chromatography? Chromatography is a method that separates target compound from other chemicals in the sample. The separation is based on the differential interaction with two chemical or physical phases. There are many forms that are based on the different form of interaction between mobile and stationary phase. We will be discussing all the difference and all forms of chromatography in this lecture. It is used for chemical analysis or to prepare specimen for analysis by other technique. So it can be like a standalone method used for analysis of a chemical, or it can be mixed, added to another method for detection. The concept of chromatography came from 93, where they found that the plant pigment can be separated in this form. It can give different colors, and the word graphene is to write, so the word chromatography was described for the first time. To know the basic principle in short and brief, basically we will be having a mobile phase which will travel through the chromatography system, and it will carry the sample. The mobile phase will be kept in a reservoir, and the sample will be pumped, and inside an injector together, the injector will inject it into the chromatography system, where there is a stationary phase. The stationary phase is, from its name, it does not move. It is held within the chromatography system. So the sample will pass through it, and the sample component will interact with the stationary phase. Some of the sample component will be highly retained, and it will move slowly, and some will weakly interact and move faster. So this is usually in a column. The result is separated sample that will elute outside the chromatography system. It will come into a detector which will generate electronic signal, and the data system will give meaning for these signals. This is a summary of the forms of chromatography system that we have. We can divide it based on the stationary phase as planar or column chromatography. The planar can be paper, thin layer chromatography. The column is also divided into gas and liquid chromatography, and this is based on the mobile phase. For gas chromatography can be gas solid. This is also based on the interaction. Gas liquid. The liquid chromatography also divided based on the interaction into partition, size exclusion, affinity, and ion exchange adsorption. The result for chromatography system, the electronic signal that reach the detector is plotted as a function of time, distance, and volume. This is a picture that will show the planar chromatography, where you can see the separated zones are detected here on a paper, on a layer of paper. As it moves up from the solvent, this will be explained later in details. This one can be visualized from their natural colors, or a chemical or color can be added to allow the detection of the analytes. It is used for mainly qualitative, to identify analytes or to quantify them. Here for a column system, we can see how the results are plotted as a peak signal that's described over time on the x-axis or volume, and we can measure the height, the area under each peak, and the width. Knowing the retention time for each peak, it will help identify the eluting compound. So each compound will have a specific time or volume where it can elute and detected, and it will reflect the retention and selectivity of the system. The height and the area of each peak will help us to measure the amount of compound that present in the compound, in the mixture, sorry. The width, it will reflect the separation performance or the efficiency of performance for the chromatographic system. So you can assess, is it a good system, is it separating in the right way, or there is some defect in the chromatographic system. There are different chemicals interact differently with the stationary phase, so this is how they are separated. These differences with interaction as described as retention time, volume, void time or void volume. Retention time or volume, it is the time or volume when a solute exits the column and pass through the detector. So this time or the volume can be measured. It can increase with the increasing strength and degree which the chemical interact with the stationary phase. While void time is the elution time or volume that for a compound that is not retained or that does not interact with the stationary phase. So we can also take a measure of adjusted retention measure based on knowing these values for two chemicals to be separated, where adjusted retention time is the difference between the retention time and the void time, and the adjusted retention volume is the difference between the retention volume and the void volume. So this is one form of describing the retention and interaction of a compound with the chromatography system. Another way to describe the retention differences numerically is by measuring the retention or capacity factor, selectivity or separation factor, alpha. These factors are determined by the chemical that being separated, the type of stationary and mobile phase. So what's a retention or capacity factor? It is the average time a chemical resides in a stationary phase versus the time it spends in the mobile phase. If you have a value of capacity factor of 0, it means that no binding with stationary phase and the chemical is eluted at exactly the same as the void time. The greater the interaction with the stationary phase, the longer the retention time. So you will be having a value that's higher than 0, that an actual value for it. This is how we can measure it by knowing that the ratio of adjusted retention time or volume and the retention time or volume. It's a ratio between the adjusted and the retention time. So in practice, a value of capacity factor between 1 to 10 is desirable to provide reasonable separation between compounds. The retention or capacity factor, the value of K should not be dependent on flow rate or column size. It is not related to the flow rate or column size, but it can be directly related to the strength of interaction occurring between chemical and the stationary phase or mobile phase. Also, it's directly related to the relative amount of stationary phase versus mobile phase. So here we have a measure of equilibrium constant for partition of analyte into stationary phase versus the mobile phase. Well, here is the Kd is the equilibrium constant, Vs is the relative amount of stationary phase over the amount of mobile phase. Another term that we can use is the selectivity or separation factor alpha. It describes the difference in retention for the chemical that are to be separated from each other. So it's the ratio of retention factor for the latter eluting chemical over the first one, Kb over Ka. If both chemicals have the same retention factor, then alpha equals 1, which means there is no separation. Alpha increase as the degree of separation increase. Now, the term of band broadening and efficiency. The peaks of two neighboring chemicals, they have to be sufficiently narrow to allow for separation difference to be observed. The broadening of peaks is produced by various processes related to the rate of movement of the chemical as they pass around or within the support and within or between the mobile phase and stationary phase. So experimentally, they are described with these definitions: baseline width, half height width, the standard deviation of the peak. I will not go through into the details of these terms, but these just you need to have an idea about them. These values are used to measure the number of theoretical plates length, and the height equivalent of the theoretical plate. In practice, factors that affect the chromatography system, and this is what we need to know and understand: the column length, affecting the number of theoretical plates, the particle size of support, the packed beds or tube diameter, the uniformity in size, shape, packing of the support, flow rate and linear velocity, the temperature and rate of solution diffusion, the mobile phase viscosity, the degree of compound retention, the initial injection volume, the volume of connecting tubing, detector and system component besides the column. So actually these are the factors in practice that will affect the separation and the chromatographic width. So for the chromatographic width, it represents as WB, or its height width. So this is the chromatographic width. As it becomes sharper, it becomes easier for the system to separate two peaks with similar interaction and to separate more peaks in a given amount of time. Sharper peaks are easier to measure and produce better limit of detection. So this can be measured as a resolution, which is the overall extent to which two peaks are separated. So here you can see in this figure that the resolution, it's the average retention time, the difference in average retention time, and over the baseline peak width in time unit. This is the void time. Here you can see different resolutions where the separation of peaks increase as the resolution increase. You can see here when the resolution is 0.5, increase into 1, increase into 1.5. So this is ideal and adequate to have it between 1 and 1.5. A resolution of 0 means that no separation occurred. So what are the approaches to, there are approaches to increase the resolution of chromatographic peaks by whether it's liquid or gas chromatography, by using longer column, by having gradient elution, by extending the run time. Here you can see how the resolution is changing based on increasing the efficiency here, and here by increasing the selectivity. Now, after separation, after separating the component of the mixture, we'll have different analytes that can reach the detector and can be identified. This is for quantitative measure of concentration, or it can be visually identified for qualitative identification. For the quantitative measure, we use different technique to identify the analyte. First, we can compare the analyte characteristic value to a known sample of the same compound. So you can compare the retention time, the retention volume, or the retention factor to a known sample of the same compound. Or we can use a reference compound that will run in parallel in another column with having two detectors, and we compare the result between the known and the reference, also using the same characteristic value of retention time, retention volume, retention factor, to compare between the unknown and the reference. Another method for detection is to provide structural information on the analyte. It's when we are adding another method of detection to the chromatography, example of MS, like LC-MS as an example. And last method is to measure for peak areas or peak height with using internal and external calibration techniques. So Using an external calibration technique, it means that you can use a standard solution containing known quantity of the analyte that you want to detect. You will process it and separate it same as the sample, and it will usually contain one or more of specific analytes. So you will generate a calibration curve by using the standard, and by plotting the peak height or peak area versus the concentration or mass of the analyte, you will generate a calibration curve, and then you will run your sample and you can compare the results with the calibration curve to detect the desired analytes. This is the use of the external calibration. For internal calibration or internal standard, it is used to measure for peak areas or peak height with internal and external calibration technique. It is a constant amount of different compound added to each standard solution, so it will be constant, and it can be added even before the pre-treatment step if there was a pre-treatment step in the analysis. It has a similar chemical and physical properties to the analyte, and it can be measured independently from the analyte. So here where you can see the internal calibrator, and here with the sample and the external calibrator. The advantage of it, it will help normalize the result for any variation that may occur during the pre-treatment steps or during sample or standard injection into the system. So you can be sure that the analysis, the results are okay and adequate for interpretation. Now, again, these are the forms of chromatography. We will go through them one by one in brief. If you need to have more details about each technique, you can always refer to the textbook and read more about it. So for gas chromatography, from its name, the gas chromatography use gas as a mobile phase. Usually they use inert gas that has a low density, for example, like the nitrogen, helium, argon, and gas that has a low mass, like hydrogen. The gas will not interact with the gas chromatography column. It will only act as a carrier for the sample. The solution separation are based on the difference in compound vapor pressure and different interaction with stationary phase. So this is about the gas. Gas chromatography can be separated as gas solid or gas liquid based on the stationary phase. For gas solid chromatography, the same material that acts as a stationary phase can act as that act as a support. It's the stationary phase. We can add chemicals that will be retained by adsorption in the support and will allow different interaction with the stationary phase. For example, silica can be added, alumina, molecular sieves, and will have like porous materials. So the factors that affect the retention in this kind of chromatography is the surface area of the column, the size of pores in the support, the types and functional groups on the surface of the support. The other way of interaction is between gas and liquid, where stationary phase is a liquid placed as a coating or layer on the support. All these liquids have low volatility and to allow them to stay within the column. Over time, some of this liquid will leave the column. This is the concept called column bleed. This is something you can read more further about it. And this will cause the detector to have high background or to be noisy. So here maybe it's the time to change the column and replace it. This can be also minimized by using a bonded phase by reacting functional group bonded phase chromatography. So I'll show you this picture to see the difference between adsorption, where the particle can stick on the surface of the phase, and absorption, where there is an absorption. So usually the stationary phase, it's adsorbed on the surface of the support medium. Now these are the types of stationary phase that can be used in gas chromatography. You just have to have an idea about them and how they have different polarity, or can be polar, non-polar, or have intermediate polarity. This is the instrumentation. You can see that we have a carrier gas source, and it will be taken to the chromatography system. Here the samples are injected and they go back through the columns. Here we have an oven that will maintain the temperature. The control of temperature is very important in gas chromatography, and how it can elute the compound based on specific setup of temperature. Here is the detector that will take the data and will transfer it to the computer or control system. So all these part of instrumentations are important to get to know the carrier gas, how it flow, the idea of having molecular sieve beds and inline traps to maintain pure gas, to maintain as much as clear system, how we inject the sample, the derivatization of the sample. You have to know the common problems and the solutions in the gas chromatography system, the types of column and supports, as I mentioned before, the types and component of chromatography. There are different types of gas chromatography. You have to know the advantages and uses. And as I told you, if you take it from practice experience, you will learn it better. The temperature control, how it affects the elution of the sample, and the types of detectors. Now I'll talk about the liquid chromatography. As we said, we are separating them based on their stationary phase. The advantage of liquid chromatography over gas chromatography is the ability to work directly with liquid samples. It is common in clinical labs. It takes wide range of separation mechanism, stationary phases, solvents, and detectors. So this is the types of liquid chromatography. The first one is the adsorption chromatography, here where the sample is passing and some component will adsorb to the stationary phase. As I show you the concept of adsorption, it just interacts with the solid phase. It is retained on the composition, based on the composition of the analyte with the mobile phase, and bind to the surface of the support. There are degree of retention. It will depend on the binding strength of the chemical or the support. Binding strength of the chemical to the support, the surface area of the support, the amount of mobile phase, and the binding strength of mobile phase to support, which we call it the allotropic strength. Now the adsorbents can be polar acidic, they can be polar basic, or non-polar. The different types of adsorbents depends on which analyte you want to separate. This is how you will choose it. So the second type of liquid chromatography is the partition chromatography. Here is separation is based on partitioning of chemicals into a layer of stationary phase. This is usually a liquid layer. It is coated with liquid or bonded into a solid support. The solutes are separated based on partitioning, relative solubility between two immiscible liquids, between the mobile phase and stationary phase. It is divided into two forms. It can be either normal phase or reversed phase, and this is important for you to differentiate between the two. The normal phase, the stationary phase, will be polar, and the mobile phase will be relatively non-polar. In the new textbook, they are describing something called weak mobile phase or strong mobile phase. The weak mobile phase is a mobile phase that have low solvent polarity, and strong mobile phase, it will have high solvent polarity. So basically the polar compound will have the highest retention in normal phase. Okay. Example of uses is for steroids and for sugars. In reversed phase chromatography, the nonpolar stationary phase, the stationary phase is nonpolar and the mobile phase is relatively polar. So here it's the opposite. The weak mobile phase will have high solvent polarity, but the strong mobile phase will have low solvent polarity. So the nonpolar compound will have the highest retention. it will bind to the stationary phase. Example of uses is drugs, amino acids, peptides, proteins, carbohydrate, lipids, and bile acids. This is the different type of elution that can be used for different chromatography, not only for the liquid. There is the term called isocratic elution, which is the same mobile phase used through the separation. And there is a gradient elution if the mobile phase varies over time. This is done by different programming. So either by changing the solvent component through the analysis, changing the temperature, changing the polarity, there are different manipulation can allow this gradient, and this is to have a better retention of solvent of the compound. Now, the third type of liquid chromatography, stationary phase, is the ion exchange chromatography. From its name that there is ions interaction on the surface of the stationary phase. There will be an exchange. So we have a cation exchange chromatography where the stationary phase is positively charged, and anion exchange chromatography where the stationary phase is negatively charged. Example of supports used are silica, polystyrene, carbohydrate-based material like the agarose, dextran, or cellulose. Here, the strong mobile phase will contain high concentration of competing ions, so it will make it difficult for charged analytes to bind. So the strong mobile phase will pass through the chromatography, while the weak mobile phase will contain few or no competing ions, and it will promote binding by charged analytes. Retention of analyte is adjusted by changing the competing ion concentration. So it will be like constant or different composition depending on how you are separating. Example of use is the analysis of amino acid, the hemoglobin variants. It can be also used for preparation for purifying proteins, peptides, nucleic acid, and also in the lab purification system we use this form of chromatography. Size exclusion chromatography, also from the name, there is separation of chemicals based on the size and ability to enter a porous support. So the porous support can have different range of sizes. If it is small, it will enter into most of the pores, and it will take longer time for a small component to be eluted, while large component will enter only few pores and will be removed faster and before the small component. Types of support, dextran, agarose, polyacrylamide, glass beads, polystyrene. This is examples of size exclusion chromatography. Here the mobile phase can be polar, non-polar. There is no weak or strong mobile phase since this separation is based on physical differences. Example is gel filtration chromatography, gel permission chromatography, and there are other types of chromatography that use this method. Affinity chromatography, here the separation of chemical is based on the interaction with biologically related binding agent. Here they will put, or they call it, they will add an affinity ligand that will bind to the stationary phase. It either are to give us antibody interaction, enzyme or substrate or inhibitor interaction, hormone or receptor, or lysine and carbohydrate. Here the column will be selective to capture the complement, and the non-retained sample will be washed from the column. The retained analyte then will be released by, there will be like another step to wash away the bound component. So it will be released later by elution buffer that will contain competing agent, or use changes in pH, ionic strength to allow the release of the bound component. It is used for preparation, analytical application, important use for isolation of glycosylated hemoglobin in diabetes. You will see this in hemoglobin HbA1c methods by HPLC. Separation of enantiomers and stereoisomers of drugs can be made by using this method. Now the last method is the mixed mode methods, and from the name that you can use different mode of separation in one column, like the hydrophilic interaction liquid, which has a features of partition and IEC. The ion paired will have a feature of partition and IEC. The restriction axis media will have a features of partition and size exclusion. So this is, you can read them and get to know each type of them. This is the HPLC system. Here you have the solvent reservoir. Here we carry the mobile phase and it will be taken by a pump to the system. Here where we inject the sample and it will enter together with the mobile phase into the column where separation occurs. There is a temperature control for the whole system, the signal taken by a detector, and the computer will analyze the results. And also the control system will control all the chromatography pattern. Okay. So for mobile phase reservoir and delivery system, sometimes they put inlet filters. This is to prevent particles entry into the system. You will hear the term of degassing or sparging, which they will cause bubbling in the mobile phase to remove any dissolved air or oxygen. You have to maintain the quality of the mobile phase as long as you can by using these ways. You can use isocratic elution or gradient elution. The pumps itself, there is different types of pumps. It can be peristaltic, diaphragm type. This is when you use a low pressure column. When you have high pressure column, you can use reciprocating and syringe pumps. It depends how you will inject the sample depending on the pressure you want to apply. And you have to consider the volume of the mobile phase, the flow rate, the pressure, the baseline noise. All will be controlled by a specific type of pump. For the injection system, there is fixed loop injector. The consideration with knowing the injection system, that it has to be reproducible, there has to be no carryover or low rate of carryover, and it will give you different volumes of range for injecting the sample. The temperature control before injection, sometimes you need to keep the sample cold. So you can put like a refrigeration in keeping the sample cold throughout the system. Sample derivatization, this is a term that's important that you understand. It is used to improve the response of a given compound for particular detectors. It's like you are adding something to the compound, so it is easily detected by absorbance, by fluorescence, by electrochemical detector. Also, it is used to alter the separation of compound by changing the structure and retention column. The derivatization can be done from before entry into the column. They call it pre-column derivatization, or after, and this is for detection, the post-column derivatization. The type of column of support is important. There are wide selection of columns. There is the inlet filter, as we said, to remove the particulate matter. Sometimes they add a short guard column, like there's a column before the column that will contain packing material and protect the actual column and extend its life. The size is important, having to have a better efficiencies and lower detection. As we said before, to have a longer length and smaller inner diameter will give you better efficiency. These are the diameters of different columns. You can see how from conventional packed column into packed capillary open tubular system, the diameter will be decreasing, and as we said, the smaller the diameter, the better the efficiency. Temperature control, it's important to determine the reproducibility and efficiency. Unlike gas chromatography, in liquid chromatography, we need to have a constant column temperature. In gas chromatography, sometimes the manipulation of temperature will help with the elution. Here it has to be constant. different techniques to control the temperature, the column chambers, water jacket, blankets, heating, cooling blocks. The detectors, we have different types of detectors here, and it's important that you know them. At the detector, there is a flow cell, which is where the sample and mobile phase will pass through, and then we'll get the signal to be detected by the analyzing system. Here are the types of detectors that can be used for liquid chromatography. The absorbance detector, it's like when you say photometer or spectrophotometers. This is important and common. The fluorescence detector, you know the fluorescence, the same method that was described before. Electrochemical detector, the refractive index detector, and mass spectrometry. I highlighted these because these are the most common and mostly used kinds of detectors. And see, the detection limit is different between one to other. So the more if you need like to have a lower limit of detection, you will use very more advanced detector. Then we come to the data acquisition and data system. It is for automation for the chromatographic system overall control. It will record the analyst and LC method, the calibration, the control samples. It will help you identify and characterize. Yani even the system can identify and tell you what is the compounds you're getting. There are libraries of spectra or other database that can be searched to aid identification of the chemicals. So by now we finished talking about the common liquid chromatographies. There are other chromatographic methods. One is called supercritical and one is the planar chromatography. The supercritical fluid chromatography, it will have a type of mobile phase that's in between liquid and gas phase. It will be in between, and this is something used mostly in experimental laboratories, not in clinical laboratories. But just to have an idea about it, we have to mention it. For planar chromatography, the stationary phase is coated or placed into a flat surface. So from the name, it's flat. There are different types. Paper chromatography, also from the name, the stationary phase is paper. And there is a layer of water or polar solvent that is coated into this paper. Thin layer chromatography, from the name again, it is thin. What makes it thin? That the material used is different than the paper. It's like silica microparticle, and there is a glass plate or plastic sheet or aluminum sheet. The high performance is extra thin. That's why it's called high performance thin layer chromatography. So it's even thinner. So to explain how in planar chromatography the sample is separated, it's very simple and straightforward. Here you have the stationary phase and it is placed on the mobile phase. Here there is a liquid of mobile phase where it will be moving all through the stationary phase. Here the sample will be injected above the point of contact with the mobile phase. So the mobile phase will travel along the stationary phase and also the sample will travel. And at certain time, we will remove the stationary phase separated from the mobile phase. Then we analyze the point as a distance of the sample, how it traveled along the stationary phase, or to compare it with the distance that's traveled by the mobile phase. This is what we call calculating the retardation factor. In paper chromatography and thin layer chromatography, usually this is used for qualitative analysis. You have very common examples of it, and we are using them in real life. Also, they can be integrated in multidimensional separation. Multidimensional separation means there are different separation methods included in one analysis, in one equipment. Here, more explanation about the retardation factor. Like here, this is the distance traveled by the mobile phase, and this is the distance traveled by analyte A. Okay, so either we can measure the retardation factor, or we have like a control, like you have a reference compound moving together with the analyte, and if you see they are matching, means it's the same compound. If they're not matching, it's not the same. And if they are matching, you want to calculate the retardation factor to confirm. Also, another way of confirmation is by direct visualization by response to UV light, if that can give you a color, or you can add a reagent or colored product, or you make it fluorescence. Okay. Sometimes we can add also radio labels and auto radiography. So by now, I gave you a full outline about all chromatography, okay, and as short as I could have done. This is examples in clinical labs and real life where chromatography is used. So I hope by next year we can take like 3 or 4 topics, or based on the hospitals, like one day we can explain Sabah Hospital what chromatography system they have, Mubarak what chromatography, other labs or the common in point of care, and we go through the same details. So these are examples for you to know. Also, there are some phrases that I have mentioned and some I didn't. Just you have to understand what's extraction, what's derivatization, what's the isomer, enantiomer, the chiral compound, the functional group. When we mention these, it has a meaning. And by knowing them, you can know which is the most appropriate type of chromatography to use. So, have any questions?